In this quick tutorial, I'll show you how to determine allele dominance after a cross. And not just dominance, but also if a trait is sex-linked or autosomal. The question reads, analyze the results in each cross and answer the questions that follow. So the questions are, what does cross 1 tell you about dominance versus recessive? And in question B, what does cross 1 tell you about the placement of the alleles on autosomes versus sex chromosomes? Now, before we begin analyzing these tables, I want you to pay attention to these two columns. Notice the words wild type and non-wild type. Whenever we use the word wild type to describe a trait, it is the trait that's most prevalent for a specific characteristic in a population. But just because it's the most popular phenotype doesn't necessarily mean it's encoded by a dominant allele. Take, for example, eye color in humans. Most of us already know that brown eye genes are dominant to those that encode blue eyes. But still, in some parts of the world, the majority of people might have blue eyes. The majority is what dictates if something is wild type, not whether it is encoded by a dominant or recessive allele. So in the population, 33 out of the 50 males have the wild type trait, whereas 17 have the non-wild type trait. Out of the 50 females that were surveyed, 31 have the wild type trait and 19 have the non-wild type trait. And in the total population, 64 out of 100 have the wild type trait and only 36 have the non-wild type trait. So you might be tempted to think right away that the wild type trait is also encoded by the dominant allele. Let's take a look at cross number 1 to determine if that's true. When we crossed a male non-wild type with a female wild type, we ended up getting the following results, where approximately half of the 53 males were both wild type and non-wild type. That number is distributed almost equally. And of the 50 female offspring that were produced, roughly 50% were wild type and non-wild type, leaving us with very equal outputs. Question A, what does cross 1 tell you about dominance versus recessiveness of this allele? As you can tell, because these values are roughly equal, we will not be able to determine if the wild type is in fact dominant or recessive. And let me demonstrate why. Let's begin with the assumption that wild type is in fact dominant. So this female has the genotype, and we'll use the letter A for simplicity's sake, that the female is capital A, and the male, which is the non-wild type, assuming that it is recessive, would be little a. Now if we do a cross here, we'll end up with offspring that are heterozygous. So according to this cross, all of the offspring would have the wild type phenotype, since they're all capital A. But clearly we don't see that happening. We see a 50-50 split. So let's change the female's genotype to heterozygous instead. Notice how this leads to a 50-50 split we see in the cross. So does this mean our assumption of wild type being dominant is true? Not really, because the same results could be found if we set the non-wild type male as heterozygous and the female wild type as recessive. Therefore, it's inconclusive if wild type is coded by dominant or recessive alleles. In question B, what does cross 1 tell you about the placement of alleles on autosomes versus sex chromosomes? Let's assume, again, that the female, whose chromosomes are XX, is dominant, and she has the dominant allele on both of her X chromosomes. The male, on the other hand, is XY, and he's recessive. If we do the cross, we would end up with all offspring having the dominant allele, therefore expressing the wild type phenotype. And clearly that's inconsistent with the data we find in this table. If we remove this allele from the other X chromosome, these are the results we would get, where half of the males and half of the females would be affected. That's consistent with our data, but only because our initial assumption makes it true. For instance, if we said non-wild type is dominant instead, where the male's X chromosome has the capital A then no males could possibly express the wild-type phenotype, and that's not the case. So because the number of males and females in each phenotype is approximately the same, it is impossible to determine if the allele is an autosome or a sex chromosome. So let's take a look at a second cross. In this cross, we'll be taking a female non-wild type with a male wild type. The only genotypes that will give us results that are consistent with this table, with this data, is if the female non-wild type had the genotype of XX and non-wild type is recessive, meaning that she does not have any alleles for the wild type trait. The male, on the other hand, is wild type. His chromosomes are XY, and the X chromosome has the dominant allele being capital A. If we do this cross, we end up with the following. So again, the female is non-wild type, homozygous recessive. Doing this cross gives us XX, where this female carries one of the dominant alleles, and this female carries one of the dominant alleles. Notice that none of the females are non-wild type, and that's consistent here. And these males both do not have the wild type trait, which is what we see in that cell right there. So what this suggests is that this characteristic is encoded on a sex chromosome, and that the only way for it to work is if the non-wild type trait is encoded by recessive alleles. If you'd like to see another example, one that's more challenging, feel free to watch question 2 of this series, which is linked on your screen right now. Thank you for watching, and if you have any questions, use our website at biology-forums.com.